Hello everyone. My today's topic is regulation of blood glucose. So first introduction of blood glucose. It is nothing but energy currency of the body. To maintain blood glucose level, it should form a narrow limits that needs effective regulation. And regulation is very important because brain requires continuous supply of glucose and maintenance of blood glucose by metabolic process, hormones and renal mechanism. So this is very important flow chart which maintains the blood glucose levels. These are all the factors that affect blood glucose levels. So first thing is diet. After digestion and absorption, it mostly enters into blood that normally maintains 70 to 110 milligram per dl. Gluconeogenesis mainly from amino acids. So gluconeogenesis you'll be knowing formation of new glucose from the liver that from alanine or pyruvate it forms glucose. So main source during prolonged starvation greater than 18 hours. And glycogenolysis third one from liver glycogen source during early fasting 12 to 18 hours. So these three factors glycogenolysis, diet and gluconeogenesis are the factors that maintains blood glucose level. So normal range is very important 70 to 110 milligram per year. And next for use oxidation, storage, glycogenesis and conversion lipogenesis. So next I want to show two important terms. One is hyperglycemia, another one is hypoglycemia. So when the blood glucose high, what happens? It forms with the help of pancreas, it forms normal level of glucose with the help of insulin. So beta cells of pancreas plays a role in order to maintain blood glucose level. Whereas in low blood glucose, alpha cells of pancreas maintains glucose level with the help of glucagon. So here insulin and glucagon play a role with alpha cells and beta cells of pancreas in order to maintain normal blood glucose level. So this is very important. A slide, I just want to give an information regarding proposal of insulin. In 1923, Nobel Prize in Physiology and Medicine was awarded to Banting and John McLeod. So Frederick Banting and Charles Best are the person who discovered insulin. And they are the person. Next, role of insulin. For 5 mark question, they'll be asking. So regulation of blood glucose, you have to tell what is meant by glucose and what are the hormones that regulate glucose level in our body. So first hormonal thing, one is insulin. In that insulin, you have to tell this important keyword, hypoglycemic hormone. Hypoglycemic hormone is nothing but in order to maintain the glucose in the blood. So by uptake of glucose in muscles and adipose tissue, by glycolysis, protein synthesis, glycogen breakdown, protein catabolism, gluconeogenesis, lipolysis, lipogenesis, and glycogen synthesis. So in this I have shown red markings which inhibits with the help of insulin in order to maintain the blood glucose level. And these are the green I have given which modulates or positively modulates the glucose level in the blood. So this is very important slide for insulin hormone for maintenance of blood glucose level. Hyperglycemic hormones are glucagon, epinephrine or adrenaline, glucocorticoids, ACTH, growth hormone, thyroxine. All these are anti-insulin hormones. So mostly for MCQs they'll be asking which are called hypoglycemic hormones and which are called hyperglycemic hormones. So you should know the difference between hypoglycemic and hyperglycemic hormones. This is extra information, processing of insulin. If you want to get full mark, you can write this. Pre-pro-insulin to pro-insulin and pro-insulin to insulin. In MCQs, they'll be asking how many amino acids composed for pre-pro-insulin and pro-insulin with insulin. So 109 amino acids for pre-pro-insulin, 86 amino acids for pro-insulin and 51 amino acids for insulin. So mainly it occurs in endoplasmic reticulum and maturation happens in Golgi apparatus as I have told it subcellular organelles. Next, biosynthesis of insulin. These are all informations needed for detailed question. Insulin, you have to write very important subtopic in the regulations of blood glucose. So I have given located chromosomes 11 and where it is synthesized. I have told beta cells of islets of Langerhans of pancreas with the pre-proinsulin 109 amino acids, proinsulin 86 amino acids, and ER as well as Golgi apparatus with the help of C-peptide degraded from proinsulin to release active insulin. Here, your human insulin contains 51 amino acids. As I have told in that diagram, if you want to show, you can show a diagrammatic presentations also. So connecting peptide has no biological activity. However, its estimation in the plasma serves as useful index for endogenous production of insulin. So C-peptide plays a role as a marker for endogenous production of insulin. And beta cells, insulin combined with zinc and stored as a granules in the cytosol, which is released in response to various stimuli by exocytosis. So zinc plays an important role in beta cells for insulin stability. I have shown A chain 21 amino acids and B chain 30 amino acids. So both the chains held together by two interchain disulfide linkages. In addition, one intrachain disulfide link. So next, role of insulin. It is anabolic hormone as it promotes the synthesis of glycogen, triacylglycerol and proteins. So concentration 40 to 50 of insulin secreted per day. These are all extra informations. If you want, you can write a normal insulin concentration. As a clinician, you should know the normal range of insulin 20 to 30 micromu per ml. So next comes anti-hyperglycemic hormones, glucagon secreted by alpha cells of pancreas with opposite action of insulin. Here glucagon on sequential degradation it releases active glucagon with the 29 amino acids with molecular weight of 3483. So normal range I have given 40 to 130 nanogram per L. Secretion of glucagon is stimulated by low blood glucose level. So you should know at high blood glucose and low blood glucose which hormones will be produced in our body. So next to glucagon, epinephrine or adrenaline. It is secreted by adrenal medulla, increases blood glucose, promotes glycogenolysis, enhances gluconeogenesis, favors uptake of amino acids. Glucocorticoids, it is secreted by adrenal cortex, increases blood glucose, enhances gluconeogenesis, releases amino acids from muscles. So shortly I have given the points or keywords of each and every hormones involved in regulation of blood glucose. So here by seeing this picture you can tell left hand side the hero comes insulin and right hand side are other hormones glucagon, epinephrine, glucocorticoids, growth hormone and adrenocorticotrophic hormones. I have shown insulin. What are the roles? In this picture alone shows regulations of blood glucose. So as an image also you can explain in the exam point of view. So what are the pathways that activates as well as inhibits by insulin and what are all the pathways inhibited or activated by other hormones. So regulation in fasting state, I want to tell two important things. One is well-fed state, another one is fasting state. So here after 2 to 2.5 hours after meal, what happens? So 3 hours, for another 3 hours, glycogenolysis will manage the blood glucose level and thereafter gluconeogenesis will take care of the situation. Here liver, important organ that supplies the glucose for maintaining blood glucose level and hormones other than insulin, opposite to insulin, it keeps the blood glucose level from falling. They are referred as anti-insulin hormones and hyperglycemic hormones. So here I have shown like same thing, same expression but in different pictures. Whatever the image you want you can select for exam point of view. So I have shown this. What are the things? So next regulations in well-fed state. Here it is subsequent to the meal. Glucose is absorbed from intestine and enters the blood. It stimulates by secretion of insulin from beta cells of islets of Langerhans. Insulin promotes glycolysis, glycogenolysis, lipogenesis and protein synthesis. So same in picturization I have explained here. You can take a copy of it. And then blood glucose level, normal thing is fasting 70 to 110 milligram per dl, postprandial 120 to 140 milligram per dl. So again, I have shown hyperglycemic hormones, what are the sources, how far it is absorbed, different pathways, how it is involved in blood glucose regulation in order to maintain hypo as well as hyper state. So these are the very important slides you can write for exam point of view. And last thing, renal regulations. We have seen liver, bloodstream, hormone regulations, and very important last thing is renal. Kidney plays an important role. And this keyword is renal threshold. So 2-mark question or MCQ they may ask, what is the renal threshold of blood glucose? It is exceeded. Tubular maximum I have given 350 milligram per minute. Renal threshold 180 milligram per ml. And after, if it reaches the maximum limit, then it leads to glycosuria, in which glucose enters into the urine. So here our important keyword is renal threshold, 180 milligram per dl. So this is more than enough, because renal mechanism, just a hint you can give in regulation of blood glucose. And glycosuria, 2-mark question, what is meant by glycosuria? You have to write it. Now urine contains about 0.05 gram per dl of sugar. It can be detected by Benedict's test. So almost glycosuria, important key terms I have given here: elementary lag storage glycosuria, renal, and diabetic glycosuria. So very important key terms, key words used for regulation of blood glucose. And this is last slide, hyperglycemia and hypoglycemia. So what happens here? Diabetes mellitus, in hyperactivity of anterior pituitary and adrenal cortex, in hyperthyroidism. Very important word you should link with thyroid in glucose level, as well as hypoglycemia, overdosage of insulin. So as a clinician, you should take care of a person, what are the drugs they are taking. So case history, as well as what are the injections they are taking, what are the milligram, the concentrations, everything is very important for you people. Hypopituitarism, hyperinsulinism, hypothyroidism, adrenal cortical hypofunction such as Addison disease, in decreased absorption like Sprue, celiac disease. So these are the terms you should cover for regulation of blood glucose.